What I'd like to do today is talk about kind of methods, as much as anything else, for measuring genome-wide selection in natural populations, and I'll focus on plant populations. That's primarily what I work on, although some of the methods certainly can be applied in animals. And so if we look generally at techniques to measure selection at the genome scale, most evolutionary biologists are focused on molecular population genetic methods for the simple reason that they're straightforward. You sequence things and look for historical signatures of natural selection. That has a lot of advantages because you integrate thousands of generations of evolution into that signal. But what I want to talk about is kind of a bit more direct ecological timescale sort of methodologies related to looking at the relationship between individuals and lifetime reproductive success, and both the phenotypes and genotypes of those individuals. And I'll contrast kind of or three complementary methods. The first looks at transplant of fully genome sequenced individuals into natural populations, followed by kind of following those individuals, looking at the development of their phenotypes and their estimates of lifetime reproductive success to directly link genotype to fitness. I'll talk about that in relation to a slightly different sort of field study where you're genotyping native plants, those that you don't transplant, but just go out to the field site and follow through their lifetimes, measuring traits and fitness. And finally, how the different sorts of data that emerge from these studies can be combined with intensive sequencing of populations through time and across space as a distinct way to measure allele frequency change, and through the combination of these techniques, draw conclusions about the strength and pattern of natural selection acting on Snips or other sorts of variants across the genome. I'm going to focus kind of on three different questions that can be addressed with these sorts of data. The first question is kind of my main organizing question for research, and that's what maintains genetic variation in natural populations, primarily for quantitative traits, especially lifetime reproductive success. So that's a very classical question, but we have kind of unprecedented levels of data to address this kind of classical question nowadays. Second, can we predict allele frequency change from measurements of fitness or from environmental variables? What we will be doing with some of these field studies is directly estimating the parameters of classic population genetic models. And if you use those models directly, can you use a prediction of delta P to produce something to be compared to an observation of delta P? And delta P, of course, is the change in allele frequency. And so I'd like to visit that in the middle part of the talk. And the third question is, one is about, how much selection is occurring contemporarily in natural populations. What is the scale? How many loci are under selection, and how strong is that selection on scales of, a few to tens of generations as opposed to millennia? Most of the work I want to talk about is on this particular species, Mimulus guttatus, the yellow monkey flower. And this is the alpine population, which we've been working on for about 25 years. The tiny yellow dots that you can see here are actually individual plants. All right, so in this population at maturity, the plants are about that high. In other words, maybe 2 to 3 centimeters in height. They often produce a single flower. And in this single population, there's routinely 300 to 500,000 individuals that are reproducing in each generation. It's a species that kind of exhibits a life history not unusual for Mediterranean climates. It's a kind of an alpine plant or montane plant that is under snow until May or early June. And you have a picture from 2007, our field season, of what the field site looks like on June 6th. And in a very short amount of time, as snow clears from the site, they have to go either from a seed or a seedling all the way through flowering and complete kind of reproductive success, setting seed before everyone dies of drought. In the summer, there's very, very little rain, particularly by July. And as snow melt kind of goes away, everybody in the population who's remaining alive dies of desiccation. So there's this kind of short window to complete the life cycle. And, it's good for us in the sense that we can measure lifetime reproductive success with a field season of 3 months. And so the first study I want to talk about was led by Ashley Troth, and that's her in the upper right. And it involves transplant of fully genome sequenced individuals into the natural population, and then we measure phenotypes and fitness. And this is a study which started when we created a set of lines specifically from the population. We started with over 1,200 distinct lines, but after making them inbred, we ended up with a couple hundred. We fully sequenced those lines and then intercrossed them to produce outbred genotypes with known sequences. And the advantage of this method is you're moving plants in there where you know the whole genome of the individuals, but you can replicate both inbred and outbred individuals and then measure their kind of phenotypes and reproductive success. We germinate them in the greenhouse and then transplanted them as seedlings over 3 successive years with the intention of measuring selection components and phenotypes on the individual plants and then relating that to the inferred genotype of these individuals based on their genome sequences. There's one key feature about this study, which is, I think, important when we're thinking about natural selection. There's, of course, a ton of work in GWAS, both in model species and in non-models. But what was important in this particular example is the population context. Since we're interested in drawing conclusions, say, about the maintenance of variability within this particular natural population that we're measuring selection, it's pretty important that the experimental plants that we're using match the field population in terms of allele frequencies at Snips across the genome. And that was done because we extracted the lines from this particular population. We were also able to confirm that allele frequencies in the lines match those in the natural population when we directly genotype individuals. Now, the experiment was fairly straightforward. Plants, as they develop, are measured for traits such as days to flower and flower size, as well as fitness components such as survival and total seed set. And we go to every single SNP in the genome, and we ask, do you affect each trait, and do you affect each fitness component, and do you affect the relationship between traits and fitness components? And the field study involved thousands of plants, but again, fitness is an inherently variable, dependent variable that has a high degree of variability, and in some cases fairly low heritability. And so our results boiled down basically to 2 categories of variant, those which were highly significant. 45 polymorphisms exceeded significance thresholds from permutation. These correspond to about 24 distinct locations in the genome. And we have a somewhat less significant set, which might include quite a number of false positives, but are useful for looking at. And these are about 1,500 sites which exhibit an effect on traits or fitness, where they have a marginal p-value of less than 10 to the negative 5 on at least one trait. So in discussing these results, it's useful to kind of refer back to some of the more classical quantitative genetic results we had accumulated over kind of the 15 years of study prior to this particular GWAS, or genome-wide association. And classical quantitative genetics studies had shown that there is kind of a life history trade-off that's present in this population and genetically based. It turns out that days to flower is a pretty critical trait, since a lot of plants never actually make it to flower. In other words, they don't reach flower before dying of desiccation. And so speed is an important thing for viability. However, we've discovered that plants that are a bit slower tend to be larger when they flower. They have greater reproductive capacity, both in terms of the number of ovules they produce as well as the amount of pollen they can distribute. And so there seems to be a speed-size trade-off, which might be related in the field to elevated viability for the small and fast, but perhaps at a cost for fecundity if you eventually do if you reach it to flower or are able to survive to flower. And so the first result of the kind of GWAS studies is the loci that we were able to map with effects on trait kind of reiterate what we had previously discovered in terms of genetic correlation. And so what I'm depicting here is for those 45 significant SNPs, we're looking at the effect of allele on days to flower, which is on the x-axis, versus the effect of that same allele at each locus on 2 different size-related traits. How large your flower is the blue points, and how tall you are the time of flowering, which is the green points. And you can see that these two things exhibit a strong correlation at the scale of individual SNPs, which parallels the kind of whole genome genetic correlation we've estimated through more classical quantitative genetic methods. And this is results to what I identified as Q1 or question 1, which has to do with the maintenance of polymorphism. Most ideas in the world of balancing selection involve some sort of trade-off. An allele which is good in one context tends to be detrimental in another. And one of the things that can underpin such a correlation or negative correlation is antagonistic pleiotropy. And what we see here is that SNPs which have a positive effect on speed, which is how fast you progress, seem to have a negative effect on size at time of reproduction. The second thing we noticed, again, because the lines are indicative of the population in terms of allele frequency, was that there was a very definite correlation between the direction of effect of an allele and how frequent it was in the population. And so if you look at the effect of the trait on, say, flower size or days to flower, the minor allele tends to be the one that is increasing flower size or plant size of flowering. In other words, at all of these loci, which we've identified, we tend to see that the big slow allele is less frequent than the small fast allele. There's a skew in allele frequencies in one direction that correlates with their phenotypic effect. And this is important when we kind of look not only at the SNPs, but kind of the joint effect of SNPs on traits and on fitness components. So this is a rather busy graph, but what it's showing you is the field results with respect to two fitness components. Viability, or if you survive to flower, is in the top panels, okay? Or I'm sorry, it's on the left-hand panels, whereas total fitness, which is survivorship times total seed set if you flower, is in the right-hand panels. And we've separated 2 years of study. The 2014 field season is at the top and the 2016 is at the bottom. And the point of this is that if you look at the SNPs, which were significant for effects on trait, and ask what is their effect on flower size, which is on the x-axis of each of these graphs, you can see that there's a relationship between effect on flower size and effect on fitness components. And it changes between 2014 and 2016. SNPs that tended to increase flower size were detrimental to viability in 2014 and also to total seed set in that year. In other words, there was directional selection for alleles that reduced flower size and also allowed you to progress to flower more rapidly. Whereas that trend tended to reverse in 2016. There was directional selection, particularly when you included seed set as well as viability for the slow fast alleles. And these results are relevant to the question of the maintenance of variability because apart from, trade-offs between fitness components, an important mechanism potentially for the maintenance of polymorphism is temporal fluctuations in selection. And there's been a renaissance of interest in this mechanism with data from a number of species, genomic data, over the last few years. And so I provide this as an example, not really so much for addressing a particular question, but how one might use genome scale data to measure selection in a natural population context. So I want to progress now to the second flavor of study. We'll come back to the first study, GWAS kind of results at the end. But this is one where we use methodology 2, which is to go out and work directly on native vegetation, not plants that we genotyped in advance and then transplanted, but instead we go out to the field and randomly sample individuals in the population. And the methods that I want to describe here are kind of an elaboration or a genomic take on a very classical technique that was developed for the study of allozyme polymorphisms, and that's called selection component analysis. And I'll take a few slides just to describe the method. And it's very much like kind of Landy and Arnold style studies where you go out and measure traits and relate them to fitness. But here we're instead going to genotype everybody and relate that to components of fitness. So I've depicted to the left a population of seedlings at the very beginning of the life cycle. And if we were to go out and genotype a large cohort of these, we would estimate the allele frequency in zygotes. Now, that's the language of population genetics. In truth, we're measuring them at a stage post-zygote where there might be, a considerable opportunity for selection between seed and seedling. But let's pass on that for the moment. We'll just consider here's allele frequency very early in the life cycle. Then we allow the plants to progress. Some will make it to flower and some will not. All right. And so this immediately subdivides the whole population into two cohorts, what we call the A and the L plants. Those that make it to flower and those that do not. And if you genotype everybody in advance, or if you go in at this stage and genotype and separate them into these two cohorts, you've immediately got two different allele frequencies at any one SNP in the genome. You have a collection of genotype frequencies in truth, but to simplify things, this episode of selection, all right, has transformed the zygote allele frequency into two different numbers. P sub A is the allele frequency in survivors. P sub L is the allele frequency among individuals that fail to progress to flower. And the change in allele frequency in the whole population is a simple function of these two numbers, which you, again, can directly estimate by genotyping individuals that you've observed to progress to flower or not. Very simple stuff. Here's a demonstration of this method applied not to my focal population, but one that's just down the road a bit, a longer lived population at Quarry, which is depicted right here. And as a test, selection component models are rooted in likelihood. And as a consequence, you identify the null hypothesis as a constraint on parameters and contrast that to the alternative hypothesis where you allow those parameters to be unconstrained. So the null hypothesis here is that there's no selection. The genotype does not affect your probability of survival. And the consequence of that is that the frequency in the A cohort is equal to that in the L cohort, which is stated here. Here's an actual depiction of the results for one particular field season in the Quarry population. And what you can look at is delta P estimated directly from the genotyping, the predicted change in allele frequency due to viability selection. And that's being plotted for about 18,000 SNPs in this study as a function of the zygotic allele frequency on the x-axis. All right, and this is to kind of ground you in the kind of data we have here. You'll notice that the points are color-coded. The red ones means we can significantly reject the null hypothesis. We have positive evidence for selection. Whereas the black points, we cannot reject the null hypothesis. And one reassuring thing about this, of course, is that most of the red points, all right, are farther away from zero than the black points. So if your predicted change in allele frequency is small, that's weaker evidence than if it is large. So that's all nice, but what good is this kind of information? Well, in isolation, even something as simple as this is interesting and can provide information, one, about the genome-wide scale of selection, but secondarily about kind of characteristics of selection. For example, you'll notice in this particular case where I've plotted the change in allele frequency versus the initial frequency, that most of your red points are up in the upper right or lower left. And what that tells you as a practical matter is that natural selection through this component tends to be favoring the more common base in this population. If the allele is initially above 50%, it's tending to increase. It has a positive delta P, whereas if it's less than 50%, it's tending to decrease. So this component of selection tends to be making the more common base even more common in this population in this year. Viability is only one component of selection. Of course, there's a lot of opportunity for differential success also through reproduction. And I'm going to kind of describe something we call male selection, which is a little bit complicated, but simple to identify in practice. Let's imagine we now focus in an individual in a generation on only those individuals that survive to flower. Okay, those are the guys who can possibly reproduce both through male function and female function. Our population is monoecious. Every individual is both male and female, but individual reproductive success then depends on two things: the number of seeds you produce as a maternal plant and the number of pollen grains that you export, what we call outcross siring success. estimating success through female functions is quite straightforward, and a lot of people work on plants for this reason, if nothing else. And that is, the plants stay still, you follow them, and at the end of the generation, you can count up the number of seeds they produce. So you can do a pretty decent job, particularly in an annual plant, on lifetime reproductive success through female function. Male function is harder because we routinely fail to see all of the siring events as they happen. Pollen is quite small, it's hard to track, it's carried by insects, etc. And so the method here to try to detect selection through differential male success is we genotype all the moms, and then we collect their seeds, and then we genotype a subset of their progeny. In other words, we'll grow up seeds in the greenhouse and genotype those. And I've got a kind of a sampling of progeny genotypes here in the middle. And if you know the maternal genotype, in this case, say is AA at a locus where we have two alleles, A and B, and these are the four progeny genotypes, we can infer that the male must have given you A for these two and must have given you B for these two. In other words, we don't know who dad actually is, but we can infer by subtracting out mom what their contribution is to zygotes, in this case to seeds. Now, of course, when mom is a heterozygote, this becomes a bit uncertain, but it's very easy to fit that into a likelihood model and average across uncertainty, which is one of the great advantages of using likelihood models in this context. And so male selection, all right, is basically measured as the difference in allele frequency between all successful gametes, all male gametes that actually make seeds, and the total population of males that could have produced them. Now, in a dioecious species, that would be the entire adult male population, but here it's just the entire adult population, since everybody is both male and female. If there is no differential male success at a particular locus, then P sub S should equal P sub A. There should be no difference, and predicted delta P is again 0. Whereas if a locus affects male success, the favorable allele might be elevated in seeds relative to what you'd expect based on the frequency of the allele in all potential males, and this would produce a positive delta P. What causes male selection? Well, what we normally think of in this context is that some males would have greater kind of mating success than other males, the kind of idea being sexual selection of some form. That's certainly one important cause of male selection. If that's true, if we see that a locus affects the number of offspring sired, that doesn't necessarily mean it's distinct from or even different from selection through female function. They could kind of parallel one another. But the other thing which is important to consider in this term male selection is that it's not limited to differential success of the adult diploid plant through siring. This term, this component of selection, would also absorb any selection that's happening at the gametic scale. In other words, things like pollen competition or an animal's sperm competition, or even meiotic drive would kind of show up as a gametic selection and thus as a potential cause of male selection. So it's kind of an aggregate of what are in truth different components of selection that cause some males to contribute more gametes to the next generation than others. I'm going to talk about Mimulus, but I wanted to do a detour to one other study just to kind of suggest that in some situations we can parse out the different causes of male selection. We have another study done on a species called Silene latifolia, which is dioecious, where you have both males and females. And I kind of collaborated with some people on Silene because it's very different than in Mimulus, in that my Mimulus populations have hundreds of thousands of individuals. So it's quite impossible for me to genotype everybody. But we could find a Silene population in Virginia, which only had about 1,200 individuals. And in that case, we were actually able to genotype everybody. And if you are able to genotype everybody in the population, you not only can estimate male selection, you can estimate selection directly through paternity by identifying the particular sire to every seed. And that actually allows you to disentangle differential siring of diploid adults from subsequent gametic selection. And our results from the study, they're kind of not quite published. We hope the paper will come out later this year. But anyways, we do find a strong positive correlation, but it's asymmetric between paternity selection and male selection. In other words, every single SNP that showed a significant effect on paternity showed a parallel positive correlation with its effect on male selection. But the reverse wasn't always true. Okay, and that makes perfect sense because differential paternity is a subset of male selection. In other words, if you have differential paternity, that would show up as part of male selection. But if you have, pollen competition, that necessarily won't. Or it could, but it doesn't have to be positively correlated with differential paternity. In our studies of Mimulus, we really can't do paternity just because it's impossible to genotype even 0.1% of the plants in the population because there are hundreds of thousands of individuals. And so what I want to describe now is the main study we've done using the selection component technique on our main focal population over a couple of years. And this figure kind of illustrates the experimental design. In 2013, we went out to the population and marked a whole bunch of individuals that made it to flower and took, counted their seed and then genotyped in the greenhouse a random selection of progeny from each. We then went out the next year to the natural population, followed them again from the seedling stage here, which parses the entire population into two groups as you go through the 2014 season. Those who make it to flower, P sub A. Those who fail to flower, P sub L. The distinction between the brown and the green here would be an effective viability selection on a particular locus. But then, among this class of individuals, we also conducted the test for male selection by doing a second round of progeny grow-ups and genotype. I want to first discuss the results of this study in terms of question 2, which was, can we use measurements of fitness in the wild to predict allele frequency change? And then go back to questions 1 and 3. And so first, kind of the objective here for this part of the study was to measure selection in 2013 and see if we can predict the observed change, which we then estimate by going into the next year. And this is an example of what I would call prospective or forward time prediction. This is a very general thing in science, although not typically used in evolutionary biology, where you estimate the parameters of a mathematical model that predict a change in some quantity, some measurable quantity X, based on information taken in the present. Now, X, in biology could be the incidence of a disease, or more generally, the location of a hurricane, or any number of things. But the point being is you have to make a prediction now based on information at hand. You test the model by measuring a future value of this quantity, and the observed change must then be independent of the predicted change. And I think this is a useful distinction, even though it seems very basic, because a lot of applications in theory, or applications of theory in evolutionary biology, actually use the observed response to estimate the parameters of the model. And this helps you oftentimes to get a reasonable fit between predicted and observed. I should emphasize that, I'm a quantitative geneticist, and prospective prediction is really not that big a deal in quantitative genetics. It's considered kind of an ordinary thing. So one place it's used is just to predict the evolution of the mean phenotype under directional selection, either in natural populations or certainly in greenhouse populations. To the left is the famous Breeder's equation, where you have a predicted change in the mean phenotype as a function of the heritability and the selection differential. And so I want to show you an example of this in our system, where basically we imposed kind of truncation selection for larger and smaller populations. And this, was a base population founded from Iron Mountain, the population under study. And the take-home message is, if we started at the beginning and we just looked at how plants, how similar are you to your siblings, to your parents, to your offspring, we can get an estimation of H squared, the heritability. Since we designed the experiment to take 20% each generation from either the top end or the bottom end of the generation, that immediately predicts S, the selection differential. So before we started the experiment, we could use the Breeder's equation, and we would predict over 6 generations of divergent artificial selection, we would change the mean by about 5 standard deviations. We then let the experiment happen. The observed change was 5.2 standard deviations. That's an error of about 4%. All right. Now, 4% isn't perfect. I'd be happier if it was 1% or less than 1%. But it's basically fully consistent with the model, because when you estimate the parameters of a model, those are encumbered with error. I don't bring this up because it's extraordinary. I bring it up because it's kind of ordinary in quantitative genetics to kind of estimate parameters and project evolution and then see how well the projection matches what you see. Here's the application or the attempt to apply kind of the same sort of reasoning to allele frequency change from 2013 to 2014 in the selection components study. And so we're looking particularly here at male selection in 2013. And from that, we develop a prediction of change, which is on the x-axis in this graph, and the observation of change, which is based on distinct genotyping of plants from the next generation. And this, of course, depicts only a tiny fraction of the Snips that were in the study. Mimulus guttatus has ridiculously high nucleotide variation. It's kind of modestly famous for having very high pi or nucleotide diversity. And so what we've done to make a comprehensible graph here is I took all of the genes which showed at least one test with a p-value of 10 to the negative 5 or less, and I thinned it to the most significant SNP. And that's how you got a point on this graph. And for each of those SNPs, we compared the observed change to the predicted change. And this scatter plot, because there's a positive relationship, suggests some degree of success in predicting change at the allele frequency level. In other words, in predicting delta P in a manner that is analogous to what we do for quantitative traits in the prediction of delta Z bar. Now, the paper here just came out earlier this year, and a great deal of that paper is committed to kind of the statistical issues with large data analysis and how we might verify that the apparent prediction here is correct. And so here's one of the kind of tests we did, which was the method of cross-validation. In other words, you split the data set in half and use the left-hand side to make the prediction and the right-hand side to do the observation, and vice versa. There we have a couple of analyses like these, and I'm glad to discuss any of the nitty-gritty on this with anybody who cares. But the upshot is we think the prediction is quite real and significant, although perhaps imperfect. In other words, we might be underestimating at these SNPs we've ascertained the predicted change as a function of the observed change. There's a number of reasons why that might happen. So when describing this kind of experiment to people just in conversations, I get kind of two standard responses. And the first and the most common is that that'll never work. Delta P is just too small. In other words, changes at individual loci per generation are very small, and they're basically too small to detect. Now, if that were true, there really wouldn't be much point in doing these kind of direct studies where you're measuring individual genotypes and relating them to reproductive success. I'm sure it's true for many SNPs. In other words, I'm sure that many SNPs are under selection that is too weak for us to detect. But I think there are a substantial, non-trivial subset of polymorphisms that are yielding a signal large enough as long as you're going to push your sample sizes into the thousands. After that, from a slightly distinct group of people, not that it's impossible, but that this was just inevitable. I mean, how could you fail at this? Because math just works. And I think the logic here is that what selection component models are is they're just simple population genetic models where you've estimated the parameters and done a prediction. And as long as the math is correct, you can't possibly fail. I guess my response to this is I believe in math and think that math is right. But it turns out that the application of math to a natural system involves a lot of assumptions. And I think there are quite a lot of assumptions that I had to make in order to say that the predicted allele frequency change would match the observed allele frequency change. And I've highlighted a few of them here. And one of the most important is something we always skip over, and that's that, we did fully random sampling of both years in terms of both offspring and progeny and so forth. And that's a kind of one that causes failures of prediction, famously in things like elections, but which in most evolutionary studies you wouldn't actually get checked on at all. And so that kind of even fairly subtle non-random sampling could produce deviations. And also things like, I assumed the population was closed. In other words, everybody in 2014 was an immediate offspring of the plants that were present and flowered in 2013. That could break down due to things like migration, or if there is a seed bank in this population. We have no evidence of a seed bank, but if there is, that could certainly produce a deviation between observed and expected. So the second thing I want to comment on from this particular study is how it bears on the other questions, not necessarily prediction, but inference on the balance of evolutionary forces that maintain polymorphism and on the scale of genome-wide selection. And for these second and third questions, we're going to look not just at how well selection in 2013 predicts change into 2014, but also the selection that was observed in the following year, 2014, both through viability, and there was abundant evidence for viability selection, as well as male selection in that follow-up year, 2014. Male selection turned out to be considerably weaker in 2014 than in 2013. Bearing on the genome-wide scale of selection, this is a dot plot, one of these Manhattan plots. This is a genomics talk, and it is a rule in any genomics talk that you have to see one of these things, where you scale along the genome from left to right. Mimulus has 14 chromosomes. And for 4 different episodes of selection, 4 different tests, we're going to focus on the top 3. Male selection in 2013, viability selection in 2014, male selection in 2014. We've tested each SNP for evidence, and you can see, particularly in the top panel, that there are a lot of green and red dots, indicating that evidence for selection is genome-wide. There are many, many loci that apparently are influenced by some component of differential fitness in these field studies. Regarding the question of the maintenance of variation, we'd like to look at the relationship between selection at the same SNP through different components of selection. And so the next couple of plots I'm going to show you look at the relationship between different components of selection. So what I've depicted here in the upper left is the relationship between male selection in 2013. So on the x-axis here is the predicted change in allele frequency due to male selection in that year versus viability selection in the following year. And this considers all of these points are SNPs that show evidence, the best evidence for a combination of selection, a combination of evidence for selection in both years, although not for any particular direction of selection. And what you can see is that there's a strong negative relationship between male selection in 2013 and viability selection in 2014. Alleles that tended to increase differential male success in 2013 tended to reduce the probability that you would survive to flower into the next year. And this, in some sense, is completely consistent with the big fat or the big slow, small fast pleiotropy that I mentioned from the GWAS study. If it is true that siring success is related to how many male gametes you're producing, and that's related to flower size and the number of flowers, we expect those alleles to be favored by male selection, but certainly disfavored by viability selection because they tend to have an effect of slowing you down. It might make it much less likely that you even make it to flower in the following year. So this negative trade-off here is an example of antagonistic pleiotropy at the scale of individual SNPs. If we go to the lower left here, what we're seeing is the relationship between male selection in 2013, male selection in 2014. And here we see a positive relationship. In other words, the same alleles, and again, it's, this is focusing in on the subset of SNPs, which showed evidence across years. If you increased male success in 2013, you tended also to do that in 2014. So there was consistency in the direction of selection for this component between years, although overall the strength of male selection was much weaker in 2014 than in 2013. Now these two kind of imply the last one, which is that there should be a negative trade-off between male selection in 2014 and viability selection in 2014. I have grayed that out because this is a more statistically ambiguous categorization than these two. These two are based on completely different sets of data to produce the X and Y coordinate delta P values, whereas these share a value. And as a consequence of that sharing, a negative correlation is built in here. And so it's a bit ambiguous, although I think beyond that statistical issue, there's still a negative correlation. And so these all bear on question number 1, which is how is polymorphism maintained in natural populations? And this is what I was supposed to say on this slide, but actually said it on the last slide. So one of the things we can do, which is kind of a synthesis of results between these two studies, is we can take all of the genome sequences we collected for the GWAS study and analyze those in relation to this second field study of natural selection. And what we've done here is we've taken the 187 lines from Iron Mountain and done basic molecular population tests. And we can do that on windows all over the genome and so forth, and measure things like the number of polymorphisms, S, within a genomic window, the amount of nucleotide diversity within a genomic window, Tajima's D, or the Z statistic, which is a measure of local haplotype structure. We can do all that stuff, and without any consideration of the field work in 2013, 2014, we could test against neutrality. But what I'm doing here is I'm subdividing the genome into 2 categories of loci. Regions of the genome which showed evidence for male selection in 2013, and those are the pink distributions, and the rest of the genome, which is the non-significant regions for male selection, which are the blue. And what you can see in this contrast is that loci which showed evidence for selection tended to have elevated nucleotide diversity, elevated variation relative to other spots, a more positive value for for Tajima's D, and also more substantial haplotype structure measured by Z. And so these are basically just standard molecular population genetic tests. But the idea being that if a locus is under selection kind of on ecological timescales, that might be a balancing selection mechanism, we'd like to see if it exhibits the signatures of long-term balancing selection. And while these differences that you're seeing between the blue and the red might be fairly subtle, they are quite significant on the scale of 587 tests, and they're in the direction predicted by the long-term maintenance of polymorphism as predicted by balancing selection. So to summarize this part of the talk, check where I am with time here. Hundreds of Snips experienced male selection in 2013 with 1 allele at each SNP elevated in frequency among successful male gametes relative to the entire adult population. In 2014, allele frequencies at these SNPs consistently shifted in the correct direction, and antagonistic pleiotropy and fluctuating selection are indicated as potential balancing mechanisms. And so I want to sum up with just a pointer to this last method and how it could be used in combination with the previous 2. And that's intensive population sequencing, both of populations in space and of the same population through time. And of course, population sequencing across space is basically just molecular ecology. And so in the present context, it's interesting because we can classify loci as whether they were significant in the GWAS or significant in the selection component studies, and then look to see if that affects or is related to, say, divergence across space. And the one example I want to show you of this is a contrast of the Iron Mountain population where we did these studies to a population that's not too far away at Quarry. And what we find in this is by looking at divergence in allele frequency between these populations, we see that it's actually strongly related to the estimated effect of the allele on phenotype within Iron Mountain. Okay, and so what you're seeing is a divergence in allele frequency between populations related to an estimate of SNP effect within a population. And what we're seeing is a fairly strong positive relationship such that alleles that are inflated in Quarry tend to increase flower size, and those that are less common in Quarry tend to reduce flower size. Why is that relevant? Quarry has big flowers relative to Iron Mountain. In other words, the divergence in phenotype between populations is predicted by allele frequency divergence and estimates of phenotypic effect. Consider the second thing, which is sampling a population through time. For that selection experiment we did on flower size, of course, we measured the phenotypes, but as many people here in Vienna do, we whole genome sequence the ancestral population and the derived population. And what we found was that divergence between the populations in terms of allele frequency is well predicted by our estimates of effects in the GWAS. In other words, if an allele tended to increase flower size in the GWAS, it tended to become more frequent in the population we selected for larger flowers, and the reverse happened in the small flowered population. This sounds like it's trivial, but it's actually not. This is kind of failed to obtain in some studies of fruit flies where they've kind of looked for a similar signal. My point is more that, again, bringing together multiple pieces of data, distinct data sets, can provide you with novel insights, or at least confirmation. This is just me saying that in red type. So to sum up, one thing that I would say is that we've been doing these field studies for 20, maybe 25 years now, and they all have different purposes. In other words, sometimes we're transplanting genotypes with different flower size. Sometimes we're looking for epistasis. Sometimes we're studying mating system. But every time we do one of these field studies, oftentimes with transplants or with native vegetation, we do it over multiple years, the same experimental design applied multiple years. And what we always find is that the direction of pattern of selection changes from one year to the next. It doesn't matter what our purpose is. This is what we always seem to find. And so what I'm currently doing is a large-scale temporal study of the population from 1998 to 2021, where we take large collection of seeds that we've collected from adult flowering plants, tens of thousands of seeds, and sequence those as a pool to kind of estimate allele frequency through time. Now, this is a study ongoing. I'm hoping to finish it up later this year. It's the largest sequencing study I've ever done. And so anything I would say would be preliminary. But the preliminary data analysis suggests that about 95% of the genome shows little or no change. In other words, all the way from 1999 or 1998 to 2021, there's essentially zero change, or the change is so small it can't be distinguished from zero. And that's kind of what you expect in a very large population. But about 5% of the genome shows very clear evidence for greater change, selection-driven change. And the dominant pattern is not that one base becomes consistently more frequent over the interval from 1998 to 2021. Instead, it's of strong temporal fluctuations in selection. So returning to these things, these three methodologies I mentioned, I think they're interesting and complementary. You have certain advantages with method 1, the transplant, that you don't have with method 2, and vice versa. And the third one is not really an active, kind of method at the individual level, but it can provide key corroborative evidence to patterns that emerge through direct studies of natural selection. And so to finish up, I'd like to thank my collaborators on this work. Ashley Troth was the kind of leader of the GWAS work. Patrick Monahan, Jack Galicio, Julius, Leela, and John have worked with me years on the other kind of field studies up at Iron Mountain. And with that, I would be happy to take any questions if you have them and if there's time.